Hello friends, in the last video we have started with phytohormones or plant hormones. In this we discuss about growth of plant, then how it is regulated. We also discuss about 5 types of plant hormones. Then we started with auxin, how it was discovered, then the structure of the auxin and physiological features. You can refer my video, the link is given in the description box. And today we are going to discuss about biosynthesis of auxin. Sites for the auxin synthesis are shoot apical meristem, young leaves, developing fruits and seeds. Now the pathways of auxin biosynthesis. There are 2 pathways of auxin biosynthesis. First is tryptophan independent pathway TIP and the second is tryptophan dependent pathway, that is TDP. Tryptophan independent pathway does not involve tryptophan and its mechanism is not well known. So in this video we will mainly discuss about tryptophan dependent pathway. In this pathway the precursor is tryptophan. It is synthesized by shikimate pathway in plants, but this pathway is not present in animals. Now in the tryptophan dependent pathway, 4 types or 4 possibilities are there. One is IPA, that is indole 3-pyruvic acid pathway. Second is TAM, that is tryptamine pathway. And third is IAN, indole 3-acetonitrile pathway. Fourth is IAM, indole 3-acetamide pathway. In this, the first 3 pathway, IPA pathway, TAM pathway and IAN pathway, these are present in plants or these are used in plants. But this IAM pathway, it is used in bacteria. Example, agrobacterium. Now let us discuss about these pathways in detail one by one. First is IPA pathway. Tryptophan undergoes deamination in the presence of the enzyme tryptophan transaminase and it is converted into indole 3-pyruvic acid, that is IPA. Then this IPA undergoes decarboxylation in the presence of the enzyme IPA decarboxylase and it is converted into indole 3-acetaldehyde. This indole 3-acetaldehyde then undergoes dehydrogenation in the presence of the enzyme indole 3-acetaldehyde dehydrogenase and is converted into indole 3-acetic acid, that is naturally occurring auxin. So here the first intermediate of the pathway is IPA, hence it is known as IPA pathway. Now second is TAM pathway. Here tryptophan undergoes decarboxylation and it is converted into tryptamine, in short TAM. Then this undergoes deamination and is converted into indole 3-acetaldehyde. This indole 3-acetaldehyde undergoes dehydrogenation in the presence of enzyme indole 3-acetaldehyde dehydrogenase and is converted into indole 3-acetic acid. Here the first intermediate is tryptamine or TAM, hence it is known as TAM pathway. Third is indole 3-acetonitrile IAN pathway. Here tryptophan in the presence of certain enzyme is converted into indole 3-acetonitrile IAN. This is then converted into indole 3-acetic acid in the presence of the enzyme nitrulase. Here the first intermediate of the pathway is indole 3-acetonitrile, hence it is known as IAN pathway. Now IAM pathway. Tryptophan in the presence of the enzyme monooxygenase is converted into indole 3-acetamide IAM. This is then converted into indole 3-acetic acid. So here first intermediate is IAM and hence it is known as IAM pathway. This type of the pathway is used in bacteria. Example, agrobacterium. We can show all these pathways briefly here. This is the first pathway, this is the second pathway, this is the third pathway, and this is the fourth pathway. All starts with tryptophan. In the first pathway, the first intermediate is IPA. Here the first intermediate is TAM. Here it is IAN, and in fourth it is IAM. And the product that is obtained through all the pathways is IAA, indole 3-acetic acid, which is naturally occurring auxin. Now let us see auxin transport. So transport of auxin occurs unidirectionally. That is, it is predominantly polar. It is an active process because here metabolic energy is required. And why metabolic energy is required? Because it is not simple diffusion. It can occur against concentration gradient also, and it involves activity of living cell. Hence, energy is required, so it is an active process. In shoot, it is polar and basipetal. That is, transport of auxin takes place from shoot tip to roots, or in other words, apex to base. And it occurs through vascular parenchyma tissue. In roots, it is polar but equipetal, that is from base to apex. Polar transport is inhibited under anaerobic condition. It is also temperature sensitive. Some of the metabolic inhibitors of auxin transport are 2,3,5-triiodobenzoic acid, TBA, and naphthyl acid, NPA. This is all for today's video. In the next video, we will discuss about auxin signaling. So stay tuned. If you like this video, please hit the like button, share it, and subscribe my channel. Thank you.